Hey folks, today we're going to talk about mitosis and meiosis, which are two very important types of cell division that occur in our bodies. The purpose of mitosis is to create identical cells for growth and repair. So anytime we damage our cells or need to create new cells to grow, we make them through this process called mitosis. When mitosis happens, we make two new cells that are identical to each other. They're called diploids because they both have full sets of DNA. The steps to mitosis are prophase, metaphase, anaphase, and telophase. As we look at this animation, we can see that in interphase, we have a normal cell with a nucleus, and our DNA is in the nucleus and exists as chromatin. Then in prophase, you see the nuclear membrane breaks down, and the DNA condenses into chromosomes. Now in metaphase, you can see the chromosomes line up in the middle, and then in anaphase, they get pulled apart to opposite sides of the cell. In telophase, the nuclear membranes build back up, and the DNA turns back into chromatin. Then the cell splits into two, and that's cytokinesis. The whole point of meiosis is to create new cells for reproduction. So in humans, those are our sperm and egg cells. Each time meiosis happens, we get 4 new cells that each have half set of DNA. Since they have half sets of DNA, we call them haploid cells. Each of these cells is different from each other, and that's why we don't look like our siblings, even though we have the same parents. When 2 of these cells come together, they make a full set of DNA, and that's how an organism begins. The steps of meiosis are similar to mitosis, except everything happens twice. So we have prophase 1, metaphase 1, anaphase 1, telophase 1, then the cell splits in cytokinesis, and then our 2 cells go through prophase 2, metaphase 2, anaphase 2, and telophase 2, and then the cell splits again, giving us 4 new different cells. So here we see another cell in interphase. As a nucleus, is just performing its normal daily functions. Then in prophase 1, you see the nuclear membrane break down and the chromatin condense into chromosomes. You'll see the similar chromosomes come together and they'll swap some of their DNA. That adds to the variation of the cells, and we call that crossover. This only happens in prophase 1. Next, we have metaphase 1, where the chromosomes line up, anaphase 1, where they get pulled apart, then telophase 1, where the nuclear membranes build back up and the chromosomes go back to chromatin. The cells split in cytokinesis, and we have 2 cells that are different from each other. Now we skip interphase this time, move to prophase 2, where the nuclear membranes break down again, but this time, since we don't have similar chromosomes next to each other, they don't participate in crossover. Next is metaphase 2, where the chromosomes line up, anaphase 2, where the chromosomes get split apart into their separate chromatids, telophase 2, where the nuclear membranes build back up, and the chromosomes go to chromatin, and finally cytokinesis, which splits our 2 cells into 4 cells that each have a half set of DNA.